Deinococcus radiodurans is an extremophilic bacterium and one of the most radiation-resistant organisms known. It is currently listed as the world's toughest known bacterium in the Guinness Book of World Records. Ionizing radiation directly affects DNA structure by inducing DNA breaks. If these breaks are not adequately repaired, cell death can occur. There are several mechanisms by which a cell repairs its DNA and maintains the integrity of its genetic code. Single-strand break repair and double-strand break repair mechanisms are mostly present in the bacterium D. radiodurans. There are three major mechanisms that carry out single-strand DNA repair. The first is base excision repair, or BER, where abnormal bases are recognized by DNA glycosylase enzymes that bind to the damaged DNA and initiate a repair pathway that starts with a removal of the damaged base. The loss of the abnormal base creates an abasic site and then proceeds with the removal of several nucleotides. This is shown in the diagram as the final step of bacterial BER and eukaryotic long patch BER. Next is nucleotide excision repair, or NER, which removes DNA damage that changes the shape of DNA molecules, such as formation of pyrimidine dimers induced by ultraviolet light. Lastly, there is mismatch repair, or MMR, which corrects mismatched nucleotides in the otherwise complementary paired DNA strands arising from DNA replication errors when wrongful nucleotides are incorporated. Within double-strand DNA break repair, the prevalent mechanisms are homologous recombination, where genetic information is exchanged between two similar or identical molecules of DNA, non-homologous end-joining, which is a pathway that repairs double-stranded breaks in the DNA without the need for homologous template just by connecting the broken ends, often losing or adding a few nucleotides in the process, and microhomology-mediated end-joining, which uses microhomologous sequences during the alignment of broken ends before joining, thereby resulting in deletions flanking the site of the break. D. radiodurans predominantly relies on homologous recombination to repair its broken DNA, a process that is facilitated by the fact that this species has multiple copies of its genome per cell, and the DNA is condensed in the so-called nucleoid, which makes it easier for the homologous DNA molecules to find one another. Several possible models for DNA repair in D. radiodurans have been proposed. According to these models, upon double-stranded breaks in the DNA, broken end processing, known as end resection, is carried out by nucleases, and the bacterial homologous recombination proteins REC-A and RAD-A bind to the previously resected ends and promote strand invasion. Specifically, the preferential digestion of the 5' end of the DNA produces long 3' overhangs of single-stranded DNA that gets recognized and bound by REC-A. The REC-A-coded 3' end of the broken DNA invades the homologous DNA duplex and promotes base pairing with complementary sequences. DNA polymerase then extends the 3' ends using the homologous sequence of the intact copy of the DNA as a template. The synthesized strands are then proposed to dissociate and anneal with each other to form large and correctly reconstituted genome fragments. These fragments are then circularized by homologous recombination in the second phase of the bacterial pathway. The linear products require REC-A-mediated crossovers within overlapping homologies to mature into circular chromosomes.